the organizer, for giving me the opportunity to present our latest work on microbial synthesis of silver nanoparticles, characterization and application in environmental sciences. In this presentation, I will talk about the green synthesis of silver nanoparticles using thermophilic and metal-resistant bacterium. We'll go through the whole process from the isolation of the microorganisms to the characterization, the potential application of the nanoparticles. I will start by describing how I isolated this strain from an extreme environment, focusing on its ability to tolerate high temperatures and the presence of heavy metals. Then I will briefly show how the microbiological and molecular analysis I performed to better understand the features of this bacterium. And then I will explain how we used this microorganism to produce a silver nanoparticle in a sustainable way. Then I will present the result of the physicochemical characterization and the biological properties of this nanoparticle, and discuss these possible applications in biotechnology and other environmental fields. Extremophilic bacteria occupy a great variety of ecological niches, which often exhibit remarkable resistance to very high or low temperature, which are considered hostile for an anthropogenic point of view. These microorganisms are very interesting. So I was telling that these microorganisms, which are able to survive such extreme conditions, are interesting also because not only for knowledge of the mechanism responsible for thriving in such hard conditions, but also because they are a huge source of molecules which can be used in many biotechnological processes. For example, thermozymes, or the use of thermozymes, or the use of thermozymes in bioprocesses at high temperature has a lower probability of microbial contamination, increased substrate solubility, and faster hydrolysis rates. And for example, we explored the potential of these microorganisms as a producer of robust enzymes, and I will talk a little bit about in a few minutes. So as I told earlier, part of our research in the last year has been addressed towards the isolation of extremophilic microorganisms from different environments. Pisciarelli is a volcanic area close to our university, which is located in Naples, in the south of Italy. And this site is characterized by boiling pools and also water springs. As for other volcanic areas, this area is characterized by aggressive sulfurous effluxes with sulfuric acid is the main gas, and high concentration of some elements such as arsenic, mercury, copper, cobalt. Therefore, this area represents a big reservoir of microorganisms able to face extreme conditions like very acidic pHs, very high temperatures, and high concentration of several metals. At the time of sampling, we sampled in this boiling pool. I will show you the cursor. We sampled in this marginal area where the temperature was around 55 degrees and the pH was around 5 degrees. And we were able to isolate microorganisms which we identified through 16S RNA sequencing. The microorganisms resulted to be Alexicolobacillus mali, a gram-positive bacterium with an optimal growth at 55 degrees, pH 4, highly tolerant to arsenic. The genome was successfully sequenced, and we made a functional annotation showing a predominance of genes which are involved in many in general processes like amino acid and carbohydrate metabolisms, but we also found many other genes involved also in metal resistance. We made also a functional genomic annotation for example, to identify glycosyl hydrolases, which belong to the carbohydrate active enzyme, which are all grouped in a website, which is the Kazime. The insight into the Kazime repertoire unveiled the presence of 32 putative glycosyl hydrolases, which are involved in the degradation of polysaccharides, and we exploited them as we cloned the genes and made a recombinant protein expression for some of them, such as for a GH9 member, which was a cellulase enzyme, then active on beta 1,4 and beta 1,4 glycosidase linkages, and which is active also on filter paper cellulose, so is able to degrade cellulose. We also characterized a hendoxylanase, which is active on xylan, and a beta xylosidase, and the biochemical characterization of all these enzymes demonstrated that these enzymes can be used as active biocatalysts for the degradation of lignocellulosic biomass. And for example, we set up an enzymatic cocktail between the hendoxylanase and the beta xylosidase to produce a high level of xylose from some agri-food wastes. With a similar approach, we also identify and characterized another thermophilic extremophile, which resulted to be Geobacillus stearothermophilus. It is a gram-positive bacterium with an optimal growth temperature of 60 degrees and pH 7, and which also resulted to be very tolerant to arsenic and cadmium. Also in this case, we performed the characterization of its genome, and also gave a look at the Kazome repertoire of these microorganisms. Also in this case, we could find a less number of putative glycosyl hydrolases, but a complete set of enzymes which could be responsible for xylan deconstruction. And in this way, we assessed the new method to demonstrate that we could use the entire repertoire of secreted enzymes by these microorganisms to generate, also in this case, xylose from many waste biomasses. In more recent time, we decided to exploit the possibility to use extremophiles for the green synthesis of nanomaterials. Because their natural resistance to metals, and due to also their high metabolic versatility. It has been established that metal nanoparticle formation can be considered as a defense mechanism against toxic metal through reduction mechanisms that can lead to less reactive metal oxidation states. we also consider the fact that nanoparticle synthesis with the chemical technique may require the use of toxic reagents, while biological methods can represent more sustainable alternatives, as synthesis occurs through living organisms. Bacterial-mediated synthesis has been demonstrated to have some advantages over algae, fungi, and plant extracts, which are also used for the setup of green nanoparticles. The production of metal nanoparticles in using microorganisms or using living organisms requires milder conditions, the possibility of not using harmful reagents. And so all these reasons brought us to investigate this aspect. In this slide, I'm showing the experimental setting that the protocol that we set up to assess the formation of silver nanoparticles. So we started growing the bacterium and then collecting the secretomes. To the secretome, we added different concentration of silver nitrate, incubated for a while, and then recovered by centrifugation the nanoparticles. We made a thorough characterization of the nanoparticles with several techniques, and also assessed their biological properties with some studies that I'm showing in a while. To determine the optimal condition for the nanoparticle formation, we evaluated the effect of various parameters, such as silver nitrate concentration, temperature, pH, and time. And all the synthesis resulted to be more efficient at 60 degrees centigrade at pH 8, and in a time of incubation of 24 hours. And we also assessed the best conditions were using 0.5 and 2 millimolar silver nitrate as the precursor. The formation of the silver nanoparticles was assessed, verified, registering absorbance spectra that revealed typical surface plasmon resonance peaks with absorption maxima around 417 and 470 nanometers. These peaks, generally peaks below this value can be considered associated with the small spherical silver nanoparticles, whereas peaks above 430 nanometers are indicative of much bigger aggregates. We made a characterization through dynamic light scattering, and here are represented the distribution plots of 0.5 and 2 millimolar nanoparticles. In the table are reported the dimension that, as can be seen, the size increases with the increasing the amount of precursor, and the increase in silver molarity leads to a decrease in the zeta potential, resulting in more colloidal stability. And we also observed a decrease in the hydrodynamic diameter and the hydrodynamic parameter. The formed silver nanoparticles were also analyzed through scanning electron microscopy and transmission electron microscopy, and here are reported the results of the SEM analysis. As can be seen, both samples exhibit a subspherical shape with differences in size, while with only slight differences in sizes, we can see better from the TEM analysis, but likely a very different texture. The nanoparticles synthesized using 0.5 millimolar have a subspherical shape, and the dimension that was measured was about around 17 nanometers. We also made some FTIR analysis to try to investigate on the surface chemistry and interactions of the silver nanoparticles. As can be seen from the spectra overlaid from 0.5 and 2 millimolar nanoparticles. In the aliphatic region, some differences can be noted, which indicated variations in the organic molecules which are adsorbed on the nanoparticle surfaces. This better defined peak in the 0.5 millimolar suggests a more ordered and structured organic coating in comparison to the 2 millimolar ones. Also, when giving a look in more detail at the fingerprinting region, we could observe that at lower precursor concentration, the molecules look more able to vibrate and less densely packed than in the other sample, suggesting that there can be different coating using different conditions. The isothermal gravimetric analysis was therefore employed to evaluate the thermal stability of the nanoparticles synthesized at 0.5, starting from 0.5 and 2 millimolar precursor, and the thermograms revealed a minimum weight loss during the initial phase. Upon reaching the isothermal phase at 120 degrees, the weight loss stabilized, suggesting the absence of further thermal degradation or any significant mass loss, confirming the thermal stability of both formulations that can be ascribed to the presence of robust biomolecules, thermostable biomolecules that function in the coating of this nanoparticle deriving from the extremophilic microorganisms. After assessing that these nanoparticles can be then produced in different possible structures, considering variation in precursor concentration. We also wanted to assess their potential, and first of all, we made some characterization of their antimicrobial activity. In this slide, it is reported the bactericidal activity of nanoparticles used at a concentration of around 100 micrograms per milliliter, in which the Staphylococcus aureus was completely resulted to die after 20 hours of incubation with 0.5 and 2 millimolar nanoparticles. The Pseudomonas aeruginosa result to be much more sensitive to the treatment with the nanoparticles, as cells already died after 3 hours of incubation, as can we see in these slides, in this part. Regarding Salmonella typhimurium, they also was eliminated after 20 hours of contact. whereas the microbial load of Listeria monocytogenes was reduced by 1 log, but not completely. The cells did not completely die. And similar results were also obtained for Candida albicans, where we could observe, in comparison to the control cells, a reduction in viability, but not complete removal. Overall, these results, which are resumed in this table, show that the nanoparticles have a strong antimicrobial and antifungal potential. And we also could verify that the concentration of nanoparticles that we used were in some cases lower than those reported in the literature, thus confirming a good potential for their use as antimicrobial agents. Possible mechanisms can be related to the damage of cell membrane, to DNA damage, protein damage, on formation of hydroxyl radical oxidative stress. We also wanted to assess the antioxidant activity of the produced nanoparticles, and for this we set up DPPH and ABTS assays using Trolox as a positive control. As can be shown, we use a different concentration of nanoparticles, and could observe a dose-dependent effect, either in the 0.5 millimolar and 2 millimolar preparations. The best results were obtained when using 2 millimolar nanoparticles, and the highest concentration of nanoparticles, reaching values around 80% and 75% scavenging activity. Therefore, we also try to assess the cytotoxicity of the nanoparticles, performing a hemolysis assay using either red blood cells and also erythrocytes from individuals with beta thalassemia. We also, in this case, we tried with the different concentration of nanoparticles, and measured the hemolysis of the hemoglobin through a spectrophotometric reading at 540 nanometers after 2 hours of incubation. As can be shown in all the conditions that we tested, the nanoparticles had a minimal effect on the hemolysis. The highest values was reached with the 2 millimolar concentration, with the nanoparticles obtained from 2 millimolar silver nitrate, and the highest concentration, but these values also on thalassemic red blood cells stayed well below the clinical safety threshold of 2%, indicated a low toxic risk for these cells. We are now carrying out experiments in vivo in cells to check if there is a cytotoxicity of our nanoparticle. But these preliminary results give a proof of good hemocompatibility. Last but not least, we also tested the potential of silver nanoparticles as nanocatalysts to degrade dyes of toxic compounds. In this case, I reported the results on 4 nitrophenol, and as can be seen from this side of the slide, the only incubation of 4-nitrophenol with the sodium borohydride did not change the absorbance of the compound, whereas adding 0.5 millimolar and 2 millimolar nanoparticles, a reduction in the peak corresponding to the absorbance of nitrophenol appeared. A complete reduction could be observed, and an increase in formation in another peak corresponding to the formation of the product of the reaction, that is the 4-aminophenol, which is much lower toxic than for nitrophenol. Very similar results were also obtained. Okay, these are shown that we could observe the complete degradation already within about 30 minutes in using both kind of nanoparticles. Very similar results were obtained when using Congo red as the toxic compound. Also in this case, we could observe a lower peak corresponding to the complete degradation of the compound, but it was also visible by naked eye observation, as the Congo red is red, and the solution became very clear in half an hour, confirming the strong potential of the strong catalytic potential of this nanoparticle, which can be then tried to be assessed for their use in the degradation of toxic compounds in wastewaters. Okay, so I can move to the conclusion. We set up a green synthesis method and reported about the characterization of microbial-derived silver nanoparticles. Geobacillus stereothermophilus appears as an excellent candidate for the biosynthesis. The nanoparticles demonstrated antioxidant activity with the highest efficiency observed in nanoparticles synthesized at 2 millimolar silver nitrate. They demonstrated high biocompatibility, superior catalytic efficiency, and also antimicrobial properties. Therefore, we think we could further exploit these nanoparticles, which can be produced in a quite easy way for several applications, ranging from environmental applications to possibility to set up also application for food packaging, as they have antimicrobial and antioxidant properties. And so we are continuing our studies to further investigate the possible use of this nanoparticle. We need to understand if they have an impact on the environment, and also wanted to set up if they can, we would like to explore if they can be used for biomedical applications, as we could observe that we are able to produce also very small nanoparticles, but of course much more studies are still required. I want to thank the group which helped me, my group in Napoli, which helped me to obtain these results. In particular, Dr. Alessia Di Fraia, who performed most of the work. This work was supported by Italian funding, and I thank you, all of you, for your attention.